Donc, notre troisième conférence ce matin est donnée par un nouveau membre du CEF. Il sera donné par Mme Ilga Porth, qui nous parlera de Genomics Research on Invasive Forest Pests and Pathogens, Epidemiology and Host Suitability. On va lui laisser le plaisir de nous introduire à sa recherche, à ses travaux. C'est une nouvelle professeure à l'Université Laval. Donc, professeur Porth.
Merci beaucoup pour m'avoir donné la possibilité de parler avant vous. Je vais continuer ma présentation en anglais. So I feel more comfortable still to talk in English, although I have done some French classes and I was successful in passing them. But I feel still more comfortable in English to give you my research program. Yes, I joined very recently as a regular member the Centre d'études de la forêt, and I have been with Université Laval since around a bit more than 1 year, 1.5 years already. Time is running quite fast. And I have started writing a lot of project applications to fund my research. What I'm interested in, I'm a biochemist by training, but through my PhD and my consecutive postdocs that I did at the University of British Columbia, I found much interest into, first of all, in forestries, and second, in forest genomics. And in particular today, I would like to talk about a recently funded Genome Canada project, which is based on research regarding insect pests and pathogens. So I'm not really dealing here much with trees, just indirectly, which is something new to me. But I do bring the background of forest genomics and genetics in this program with me. So the work that I'm interested in doing and that I'm working right now is related to the X1, the genetics, molecular ecology and ecophysiology. But I think the research and the dynamic of the ecosystem, the forestry ecosystem, especially in terms of invasive species, it could be invasive plant species. I'm really interested now in ecology as well, but also in invasive pests and pathogens. I think I touch several axes here in my research. So on the left side, on this side, you see the project that I will be mostly talking about, which is related to biosurveillance in relationship to invasive pests and pathogens that are not really established but could get established, or they have been already introduced but were successfully eradicated. And the aim is now to figure out if there are any new establishments when we do some biosurveillance. This work is done also in collaboration with the Canadian Food Inspection Agency with the Plant Protection Department. And it is a multi-institutional, multidisciplinary big project. So in total we have more than 50 team members. So I'm personally at the moment more directly involved with about 6 high qualified personals. And I'm also the co-project lead for the Université Laval team. On the other side, I have still, in part, also applied for funding regarding my other interest, which is forestry genomics. I have been working extensively at the University of British Columbia with poplar trees, especially with Populus trichocarpa, black cottonwood, where we have produced a lot of genomic resources, but also we have done a lot of phenotyping. We have done a lot of phenotyping in terms of phenology, ecophysiology, growth, and also pathogen resistant, but also a lot of work regarding wood quality, wood chemistry, and wood structure. In total, we have evaluated more than almost 100 field traits actually in a common garden at the University of British Columbia. The other work that I have done at UBC, which was my first postdoc, was regarding with spruce, and I'm still related to spruce indirectly, but also directly by supervising a PhD student in the Sprucer project, and I'm working on spruce functional genomics there with a recently hired PhD student. I'm still continuing to work on deciduous tree species, namely poplar, but although I'm more familiar with working with Populus trichocarpa, Populus balsamifera is a subspecies or sister species, very closely related species. So and other species that we have here in Quebec in the east. So I will continue working on this, and one of the things I'm also interested in is tree improvement. So I've been involved also in genomic selection in spruce, for example, at the University of British Columbia. We have done here some project, or Douglas fir genomic selection. But for my recently funded NSERC Discovery Grant, I will be working on poplar again and try to. Poplar is not a domesticated species, so there's not too much breeding program going on. But I would like to use genomic tools and also the phenotypes that I have already measured to put this together in a model and then find some interesting genes which I can use then further in breeding in some advanced breeding program. And this also involves some testing, doing some transformations as well, and doing again phenotyping for stress or phenology and other indicators. And I'm also interested in structural genomics. So one of the things is to look at structural variation in the genome, for example, gene copy number variation, which has been already done for a couple of years at Laval University, especially with my colleague whom I took the place from, is John Mackay. So I'm working with a postdoc there as well, and while it was done in spruce, I'm now working on the same questioning for poplar, because this kind of structural variation are implicated in stress as well. So they could actually equip the tree or individual genotypes with some resistance or tolerance to stress. So that could be, we're looking mostly at abiotic stress, so we relate this to climate mostly, but we're also interested to look at soil conditions as well and see if those genes or if those structural variations are also implicated there in, for example, in reforestation of mining sites or something like this. If we can find genotypes also by screening those individuals. So I would like to start with with the project that I will be really talking about today, which is BioSafe. It's an acronym for Biosurveillance of Alien Forest Enemies. So it was just recently funded, but actually the official start was already the 1st of October. And so we are already way into our third quarter of the research. It was funded through the 2015 large-scale applied research project competition in the forestry sector. And the lead Genome Center is British Columbia, and the project leader is Jean from the University of British Columbia. And then we have two other project leaders. One of them is also our end user, the Canadian Food Inspection Agency, represented by Mr. Cameron Duff, and myself, and the team at Laval University. So we are really interested in outreach and engagement and communication of results, and also of the starting of our project, because I think it is very important to get people to know and make aware of the problematic of insect pests and pathogen pests. So as we all know, forests are very important for the Canadian economy, but also for the culture and the society. And the contribution to the GDP is estimated by around $22 billion. It creates in Canada around 250,000 jobs. And the issue that we are dealing with here is through all this through global trade and especially through different continents, different climates, we have an increasing threat of invasive species. And in the West, it comes definitely so from Asia rather, when we have high import volumes, and of course this poses an increased threat to basically to have invasive species established in Canada if they find appropriate host trees and if the climate fits them, the species as well. But also on the other hand, if we have problems here, we could have issues with market access. So the fiber supply could also be threatened when we have a problem, but also we could also, of course, lose forests when we have an infestation. For example, like, although it is an indigenous species, the mountain pine beetle epidemics. So where are the costs involved? So, of course, the highest costs are estimated when we do nothing. So regarding the four species that I will mention later, which are 2 insect species and 2 pathogens, the cost of inactions are estimated by $2 billion, actually. The best way moving forward is of course, to prevent that those species come into the country as well. And or also to be prepared in case this could happen. And if it happened, then to provide an adequate response. But when the damage is done, we have to think about recovery. So around the line of the budget, of course, recovery is already very costly, and inaction, of course, the most. So what are right now the challenges of forest invasive alien species surveillance? So what we need to do is to identify rapidly and accurately pests and pathogens, and this has to be done at different life stages. So in terms for insects and eggs, egg masses, as you see here. So this is an egg mass of the Asian gypsy moth, one insect that we are working with, and also adult insects or larvae. And also, for example, you need to know if there infection happens, because sometimes the infection can be asymptotic, so it is infected, but you don't really see the symptoms. And important is also, as shown here, with the checking for possible infestation in the nurseries. So these tools that we would like to develop could be used in all those cases. It is also important to track the sources of the infestation or occurrence, to identify the origin and the pathway, so how those pests and pathogens basically entered the country. And also it is important to assess the risk of their establishment. Right now, we found that there is a lack of effective tools that integrate the costs, benefits, and also epidemiological data to make evidence-based decisions. So our solution is to do genomic biosurveillance. We have identified, together with the Canadian Food Inspection, 4 top-ranked foreign invasive alien species. 2 insects, the Asian longhorn beetle and the Asian gypsy moth. And then also the Dutch M disease-causing fungus and a sudden oak death or moissates. We would like to provide genomic surveillance tools to enhance Canada's capacity to prevent and to mitigate those invasive threats. To do so, we have assembled a large team which is multidisciplinary, multi-institutional, and for each of the 4 species, we have team members. Some members are, of course, working on different insect or pathogen pests due to their expertise. So we have geneticists, people who are also familiar in particular with genomics, and pathologists, plant pathologists, and entomologists, and bioinformaticians as well. We have a large team of bioinformaticians or people related to bioinformatics. And we have also a jails team, which is the connection between genomics and society. So each large-scale Genome Canada project has to have this jails component. So these can be economists or people from the social sciences who look at the effects or the positive effects, hopefully, or what the people think about the application of genomics for example, in this case, in terms of forestry. It could be applying those tools, or for other projects, it could be to plant genetically improved stock. I don't mean genetically modified here, but genetically improved using genomic tools. And then, of course, we have also project management, who are very important to actually run the project, this big project. So as I mentioned, this is multidisciplinary. So we have entomologists, pathologists, bioinformatics, population genetics is very important for us as well, and quantitative genetics as well. And people who know all of this and work on the genomics tool development, JS people. And also we have the technology transfer component, which is directly related to our relationship with the Canadian Food Inspection Agency. So we have a number of investigators, which are those numbers, which are the so-called co-PIs, which are the bosses of the small groups. And underneath them, there's a whole armada of people working to get things done. And so we have, as I said, multi-institutional. We have university researchers, the government, the CFIA as well, and as well as an end user. We have FP Innovations as well, and we have also the Canadian Forest Service. how does now our pipeline of work look like? In order to get what we propose to deliver, namely the genomic biosurveillance tool, we have 5 activities. The first activity is related to the genomic characterization of global outbreak samples. So for all the 4 species, we have extensive sampling. We have hundreds and sometimes thousands of samples as well. And the second activity is the one that is related to doing more fundamental research, if you so will, which is applying quantitative genetics to find interesting the genetic architecture of ecological traits or epidemiological traits. For example, in the Asian gypsy moth, we're interesting to identify the genetic basis of flight capability, because this is a trait which is related to invasiveness, or other traits that are related to cold tolerance, for example, as for the Asian longhorn beetle, or pathogenicity, virulence for the Dutch elm disease and SOD as well. For southern oak death, also host suitability. And the third activity is the design of the genomic biosurveillance tools, which is a confined number of genetic information that can be used, that is so significant that allows us to predict basically from those data the sources of the infestation. And the fourth activity, last but not least, is the JAILS and the decision-making tool that people at the University of British Columbia are working with. And this all feeds into the technology transfer and implementation of the whole system. And this will be delivered to the Canadian Food Inspection Agency. So our deliverables are population-specific diagnostic markers from activity 1. From activity 2, we have candidate genes markers for epidemiology traits. And in activity 3, we generate a standardized and validated biosurveillance genomic tools. And I mentioned before, important is also to use all this information to develop a predictive algorithm to classify outbreak and service samples to the source. And the decision support tool that incorporates the socioeconomic and epidemiology data is at the core of the whole project as well. And then finally, we will generate a web portal entry for the end user. The deliverables that benefit the end users. So we will receive from the end users field samples in a way that we will test our genomic tool that we have developed by the other sub-activities. And this information will go back into the decision support tool and will be feeding the BioSafe web portal. And the information that we get there will then be communicated again to the end users, which is the CFRA, industry, innovations, as I mentioned, provincial agencies and federal departments. The project timeline is, so we are now in Q3 actually, so we are still in the discovery phase, but at the end we have something which is called a translation. So when we have our genomic database established, we started already the technology transfer. So we are working right now on developing population markers and the trait markers, which will help us to make genomics tools, and this information will be tested again. So we will get survey samples, and the results from the survey samples will feed back again into the genomics database. And in parallel, we will establish our decision support tool, which will be again fed by the results from the genomics database. And for tech transfer, we will provide the genomics tools, the genomics database, and also the decision support tool. And so this is the whole idea of our biosurveillance network. Thank you.
I thought Brian Harvey's not here, so I could ask some questions. For those who know Brian, Brian always asks questions for everybody. I was curious, just thinking about trying to make genetically resistant trees. Trees are long-lived organisms. If we developed a genetic resistance and you have an epidemic insect that there's billions of them and 0.1% are not affected by this resistance, it would seem like you could have a fast microevolution towards the insects that are able to get past the new host resistance. And I was thinking, depends, I guess, probably how many trees you plant to create this kind of ecological pressure. So there's probably some threshold under which you wouldn't cause a microevolution. And I was wondering if anybody in your group is working on that kind of thinking, how much is too much and where can we go to not create a new problem?
About having resistant trees?
Well, if you develop resistant trees, and they're going to live there for 100 years, but there's 0.1% or 1% of the insects that are able to get past that resistance, and so they're creating generations every year, and then in, say, 10 years, all of a sudden 90% of the insects are no longer affected by. How do we make sure that the resistance is really going to work in the long term and not be like in hospitals where we see bacteria getting around C. difficile, for example? So could that kind of thing happen with insects?
Yeah, the problem is therefore, but this is a multifaceted problem, I would say. Some of the problem is of course that when trees are stressed already, so let's say by climate change, so they are stressed, so they will become more preferable hosts for incursions, for invasive species. The other thing I think is also important, I would say personally, that if you plant trees that are more mixed, that are less uniform, I think it is better clone in a way that you have monoculture. I think that is a great problem as well. If they have some phenological mismatch, I think that would be good with the insect. Of course, the generation time of an insect is way shorter than of a tree, and so what we see, for example, those insects that could establish here, actually they don't cause much problem in the area where they originate from sometimes, but then when they change. I don't know why, but they become, for example, for Dutch elm disease, it is supposed to come from Asia somewhere, but there are no symptoms or anything, so basically it does not exist per se as a symptom, but at the elms there. But here in Europe, it causes a lot of damage. So, well, some people also think they can plant some, at the corridor, for example, some resistant trees, some genetically modified trees, and this will form like a barrier, and then the insect or the pathogen is unable to enter, basically, and stays there. So this somebody, for example, has also such suggestions, yeah.
Okay, so I see a multi-tiered approach.
It's a multi-tiered approach because there's so many factors. I think for me, I feel that having perfect phenology match and monoculture is the greatest threat, I think, I would say.
Thank you very much for a nice presentation. I agree totally with you that better taxonomic tools and identification tools, particularly genetic ones for pathogens, is really important. But the other side of the whole invasive species issue is monitoring. Can you comment to what extent this project will kind of be implemented on the ground? As I understand delivering tools to CFIA is important, but if we look at specific examples, most of the time our problem is falling down with lack of sufficient monitoring. You can look around the city of Montreal and the spread of emerald ash borer, for example, as a clear example of a failure in that regard. Can you at least just give us a couple comments on how the project will feed back into actual monitoring? Thanks.
Yeah, I think in the decision support tool, we have also some action happening that we would be interested basically to do citizen science, so they could basically report something, what they see. We talked about that, but this has to be, and the CFRI said that has to be again validated, but it could be reported and could be validated. And regarding, so where it could be applied, so it could be, for example, definitely the inspectors at ships would be able to apply it for the Asian gypsy moth, for example. And for ALB, this is usually found, the problem in packaging with ALB-infested poplars from Asia. And for the southern oak death, that would be applied in nurseries, for example, that tool. And I would also like to say that the CFI is not only co-PI, I mean, the representative and providers with they're really full supporters, and they already have the technical equipment who will also help them then to continue that work as well. And the whole data and everything will be hosted in a way that is accessible to the people, to the agencies who will be using them.
En fait, je voudrais remercier les trois conférenciers de ce matin qui, dans le fond, dressent un assez bon topo des principaux axes de recherche du CEF. Comme vous le voyez, nous continuons à recruter des personnes qui alimentent ces axes de recherche. Je vais laisser la parole à Luc Lauzon pour vous donner quelques instructions sur l'heure du lunch, puis le retour, parce qu'on revient en salle cet après-midi à partir de 1h30. On va être dans des sessions qui sont divisées dans 3 salles différentes. Donc, Luc, je te laisse la parole pour donner un petit peu les infos.
Alors, on a quelques informations à vous donner. J'inviterai Mélanie de venir nous voir en haut. Donc, pour le dîner, ça va se passer au SH 4800, donc c'est 2 étages plus haut. Il y a 2 choix de menus. On a une table spéciale pour les gens qui ont fait des demandes explicites pour des repas avec restrictions alimentaires. Ces repas-là sont identifiés à votre nom. Je vous rappelle qu'on a un AGA qui va commencer le temps que les gens aillent se chercher leur sandwich. Vous venez nous rejoindre au SH 2120, c'est juste ici. Il y a quelques changements à l'horaire, n'est-ce pas? Et donc, ce matin, on est obligé de déplacer la conférence, l'allocution du conférencier invité qui va la faire demain matin. On a donc demain matin, 8h30, le conférencier invité qui va nous parler, suivi de la dernière conférence du nouveau chercheur invité, du nouveau chercheur régulier, pardon, qui est Daniel Hull. Et comme on a dit ce matin, Benoît Lafleur ne pouvait pas être ici, donc c'est comme ça qu'on va faire les présentations. On va prendre un peu de temps sur la session d'affiches, mais inquiétez-vous pas, vous allez avoir amplement de temps parce que la session d'affiches, ça se déroule en même temps que le dîner. Oui, et donc les gens qui sont aux sessions étudiantes, coupe partielle, sylviculture, ainsi que les techniciens, juste vérifier l'horaire. Là, on a changé l'horaire en ligne, donc il y a un petit, on décale d'à peu près 20 minutes. On a des activités sociales pour vous, comme à chaque année. Ce matin, il y avait 4 braves personnes qui ont couru sous la pluie dans les méandres de la construction de la Ville de Montréal. Et ce soir, vers 17h, rendez-vous avec Jérémy dans le hall où on a fait les inscriptions pour vous rendre à H-Montréal. 10 minutes de marche? 15 minutes de marche. Et en chemin, vous pourrez vous acheter de la nourriture. Jérémy. Pour les autres qui n'étaient pas inscrits, où on avait seulement 60 places, donc pour les autres, on suit Mélanie, qui a réservé un bar qui s'appelle la Station des Sports vers 18h. Malheureusement, il n'y aura pas de partie de hockey des Canadiens. Les gens qui participent au lancer de la hache et qui veulent aller nous rejoindre après l'activité, vous êtes les bienvenus. Il y a aussi une conférence de notre ami Benoît Gendreux-Berthiaume à la Quincaillerie à 19h30, où il va nous relater un peu son voyage de retour d'Alberta au Québec en canot avec la famille. Les informations, il y a plus de détails sur le site web du CEF. Et c'est pas mal ça. Alors, on vous invite à aller chercher vos lunchs au 4e. C'est comme l'année passée, des sacs à lunch. Et les membres de l'AGA, donc, et les représentants étudiants et des employés, venez nous rejoindre au SH 2120. Merci. Ouais.